Yeah,
Right.
in Cape Valley, where we had a fire last year, the county fire, and
Yeah.
we're part of a Cape Valley regeneration project where we're kind of communities. We're trying to look at what we can do as a community to regenerate our sort of our ecosystem around us. So this seems, we're all very concerned about what damage has been done from the fire and how we can use that as a way of initiating activities.
Great. Thank you. Thank you. It's great to have you here.
Okay, I'm Terry. Can you hear me?
Yep. Hi, Terry.
I'm just here. I'm living in California in Butte County where a fire hit really bad last November, and two of my places burned. They both burned in 2008, but not this hot or drastic. And they're saying that we have a lot of contamination in our soil. And I saw this, so I signed up to just see what I could get, what kind of information. I really don't have anything to offer. I've just been going to the community fire meetings and join the permaculture local group to try to take some classes to learn how to bring the land back, but I haven't taken any classes yet. The first one is this weekend. So I'm just here mostly to learn.
Great. Thank you. That's really valuable. Thank you, Terry. Yeah. And thanks for raising that idea of contamination, because that's a real issue, and it's something we can certainly touch on in this webinar.
Okay. I'm interested in all of it. I just, it's all pretty new to me. The last fire, everything that burned pretty much came back. It just came back different. Like all the brush came back, and the trees were really hard to keep watered and wet and alive. I planted like 400 trees on 2.5 acres and spent $200 twice that summer paying somebody to water them. And in the end, I think maybe 12 lived. So, yeah, I just would like to know what anybody has to offer and just try to gather information, and so I can go forward. We're not even allowed back on our properties right now until the government comes and does a cleanup, and they're planning on scraping the top of the soil away and testing the site. And part of the permaculture group put up wattles, and they gave them away for free, and we were supposed to surround our burned house so that it would collect when it rained some of the runoff of the contamination, and then later those wattles would be taken away, destroyed somewhere, I guess. And then they said we might be able to bring mushrooms in later on to eat the contamination. And I'm living here in town. I'm not in the fire area, but this whole place was just dumped on, with toxins. And my friends, going to order a soil kit and see what it's like down here to even grow a garden, because people are growing things and calling it organic, but it just, 3 months ago, it went through all that smoke and rain. So I'm going to be quiet now and let somebody else talk.
All right. Thank you. I really appreciate that. And that was actually some really good insights that I think will contribute to our conversation. So,
okay.
Thanks, Terry. So when we're talking about fire impacts and recovery times, it's obviously going to be really dependent on how long that fire was in that space. How quickly is it moving through, what are the temperatures, and obviously what kind of materials are burning. I used some of the images that Brianna sent through, so hopefully that's all right. Some of these photos have probably belonged to people in this group, so thank you. A paper that's just come out looking at fire recovery in the Mediterranean found that it took over 80 years for that full soil ecosystem to recover. So it's not a flash in the pan kind of recovery. Oh gosh, sorry, I'll try not to use fire puns as we go. But yeah, so we're not looking at it being incredibly fast process. So what I really want to dig into is how can we actually enable these ecosystems to recover quickly. So how fire impacts on soil is, not only are we losing that organic matter in plant matter, but we're losing that topsoil organic matter that burns off and volatilizes. There's an increase in the soil pH as those minerals turn into inorganic forms, and an increase in electrical conductivity. So how much activity there is in the soil. You can see an increase in erosion, so soil losses, and we find that nutrient and water cycles break down, which Terry was just talking about from the last fire she experienced. There's a massive loss in that biomass and biological biodiversity under the ground. In very hot soils, or depending on the type of vegetation you have, then we can be forming hydrophobic waxes. So hydrophobic means that the soils literally are afraid of water, which no one ever wants in their soil situation. And then looking at how soil structure collapses. So that has an impact across the whole spectrum of recovery. If those soil become very compacted, how does that then impact on recovery? So I'm going to look at that. So this first section, I'm just touching on what are the different impacts, and then we're going to dig in deeper and then look at what actions can we take. So with these mineral dynamics, what you find is we see a reduction in those total nutrients through oxidization, so loss to the atmosphere, and volatilization, the transport of ashes, so nutrients being transported to other areas or even being deposited on your property. The leaching of materials and the inorganic lockup. So when these soils get very, very, very hot, they lose their carbon, their hydrogen, and their oxygen and convert into these inorganic, locked up, bound up mineral forms, and then erosion too. What you find is these anions, so these are elements that are more mobile in your soil, so your nitrogen and your sulfur, phosphorus and boron can be more sensitive to fire impact. So sometimes what we're seeing in the research after fire is that we can see short-term spikes in nitrogen with the long-term dynamics of that nitrogen decreasing. So up on the surface, some of the research has shown that those fires may exceed 1150 degrees Fahrenheit or 850 degrees Celsius right on the surface, but because of soil's ability to absorb that heat and buffer that heat, those top 2 inches really exceed 300 degrees Fahrenheit. And that's no picnic for those microbes. So in those top 2 inches, if they're exceeding 150, 300, those microbes will die. Below 10 to 12 inches, typically we're finding that the soil system is unaffected. But most of your microbial biomass and the organic matter in terms of short-term turnovers in the top 6 inches. So you see there is going to be an impact on biology. So in terms of what the impact is on microbiology, we're going to see a loss of biodiversity in those community structures. It's going to again depend on the temperature that the soil experience, but we're seeing perhaps a 50% reduction in fungal diversity and the abundance of fungi, and we'll get into why is that important. You'll see a reduction in your nitrogen cyclers. So people typically, when they think about nitrogen, maybe you think about legumes or rhizobia, or adding actual nitrogen. Most of the nitrogen cyclers in soil are the free living ones that in healthy soils are able to grab atmospheric nitrogen, pull that down into that system, and do your nitrogen cycling. So I'd like you to consider that soil systems are very much like our own human bodies. So in and on you have maybe 10 times the amount of microbiology that you do have in your own cells. So you are more of a bacteria than you are a human being. And the reason for that is that biology pretty much take care of so many of our health systems, from your immunity to nutrient uptake to general running of your systems, enzymes, vitamin production, all of that. So consider when you eat a piece of food, let's say you're eating an apple, you take a bite, you start to chew it, you're making it smaller. There are microbes in the soil that fulfill that job. So there'll be insects, macroinvertebrates, worms even, things that are going to take hold of bigger materials, break it up and make it smaller. So when you eat your apple, you swallow it and it passes through into the gastrointestinal system. In soil, that gastrointestinal system, those acids would be produced by certain types of bacteria and fungi that have some of the most powerful extractive acids. They can actually etch away at rocks and release bound nutrients. Then as that food passes through your gut system, you have a whole lot of bacteria and other types of microbes in that gut that help to break down, make the food smaller, and then that food becomes so small it can now pass into your bloodstream, and then you can take up those nutrients in that process, they're producing hormones and enzymes and different metabolites that then are what enable you to be healthy. So if you look at the human gut or the human microbiome project at the moment, they're pretty much saying that everything, maybe 80%, possibly more, of things that are afflicting human health come back to how well is this gut microbe system working. So the same system is very much working in soil and with plants. So I want you to consider that because plants can't move around, that their gut system is actually outside of their body. So they can't walk down to the supermarket and go and get whatever they need to eat. They actually need to send signals to different types of microbes to say, hey, you know, I'm under a bit of stress, can you send me some zinc? Or I really need to grow right now, can I have a bit of phosphorus? Or I'm coming under insect attack, who can help me? And what you find is these different types of microbes then respond to the plant signal. So there's this constant metropolis of activity happening around that root zone with plants signaling to microbes and microbes signaling to the plants. So very much like our own gut system. So without this system functioning, then we have impacts on plant communities. So I want to start with your bacteria and your archaea. So the archaea, they're named because, oh, I wonder if I mute that, can you hear me? So that's a video that's got sound. I wonder if I can pause it, it might be better. So this video just shows how quickly those bacteria can reproduce. So pretty much, you know, even if you're using your antibacterial soaps and you're trying to keep surfaces really clean, keep your kids really clean, the minute that antibacterial action wears off, you're going to have bacteria all over your system. They are in the atmosphere, you're breathing them right now. There's bacteria from every single environment that you can think of. So they are a big part of what creates rainfall, for instance. They're up in the atmosphere. They're in the deepest soil, the deepest oceanic channels in the Antarctic, top of Mount Everest. You get the point, right? So they are everywhere. They're the most numerous organism. They're very important in soils for disease suppression. They hold on to nutrients. So if we have a lot of bacteria in soil, what you get is bound nutrients. So they will hold on to those nutrients and they won't give them up until they die. Bacteria are important because they form what we call the fine microaggregates, so the very, very fine particles. Okay, so if we have a lot of bacteria, that can impact on the soil environment. So the soil on the left is what we would call a bacterial dominated soil. So it creates these systems which are what we call slaked soils. So you see these soils with very fine aggregates. You see that surface crusting and not a lot of soil structure, not a lot of water or air movement. So these soils become very dry. They can become very compacted, and we will start to see weedier species growing on it. The soil on the right is what we call a biodiverse soil. So it has a balance of fungi and bacteria in it, and we're going to get aggregate formation, we're going to get water and air movement through that. So our goal is, how do we take a soil that looks like the one on the left to the one on the right? So bacterial dominance after fire, what we're creating are these surface crusting, that compaction zone in the top, the tie-up of nutrients, and we also lose the predators. So in these soil systems, we have nematodes and protozoa that are grazing upon those bacteria. They are impacted in those top zones. Fortunately, they can move down. So it depends on, again, on the temperature of that fire and how compacted that top zone gets. They may be able to come back up, but it is an impact from fire. What we find though is we get a lot of bacterial dominance. It becomes what we call a germination signal. So it becomes a signal to certain types of plant species to germinate. So these will be your early colonizers. It might be cheatgrass. It might be some of your thistle species, what we call the weedy grasses. So species that are able to kind of clamber and get in there quickly, but often they're not the species that you're wanting to necessarily grow. They are, however, doing a job in terms of protecting that soil surface. So many of you have probably seen this image before, and my apologies, I don't actually know where I got it from. So some brilliant person did this image and I haven't credited them. So I'm sorry, and I will look to find the credit for it. So what you find is in that if we look at soils in terms of disturbance, so consider that this is a disturbance succession. So at the very beginning is where we have high disturbance. So something that would take a soil back to bare rock. So it could be an incredibly hot fire, it could be a volcano, it could be erosion, something that takes soil right back. So we have no soil, it's just bare rock. The first things that are going to arrive in that situation are going to be your bacteria and your lichens and your mosses, things that can actually get hold of rocks and start to etch away at it. With time, as we have less disturbance, then we'll start to get these small annual type plants. You'll have the little clambering species that I was talking about. They collect dust, they're bringing up nutrients, they're starting the building blocks for soil. Then we will start to get our more perennial herbs and our grass species. So you guys have, I imagine, quite an extraordinary amount of different types of native forbs and flowering species. They will come in, and then your grasses. Those grasses have the ability to really build soil, right? They are pumping out a huge amount of carbon out of their root zones. They're feeding microbiology. They're really developing soil. Then with time, I just realized this is not a very good image to show you because it's actually not accurate. So some of you have probably picked up on it that this is not an accurate image. Why it's not accurate is that the soil building processes, the depth of soil actually happens underneath grasses. What you'll find is as we get up into the trees, that soil profile or the topsoil profile gets shallower. So we may have more space between the bedrock, but the actual topsoil is shallow. So if you dig underneath your forested blocks now and have a look, you'll find that actually there's not that much topsoil at all underneath the trees. That building process really happens under grasslands. And then, you know, trees, a lot of that carbon's actually above ground instead of below ground. But what you find is there is a biological shift that goes from bacterial dominated to fungal dominated. So you can smell that when you're in a forest, you can see it, you'll see the mushrooms, you'll smell that beautiful ammonia kind of smell that comes off fungi. But these soils are very fungal dominated, right? And that's something to keep in mind as we go, is that if we're wanting to restore forest grounds, then we need to be looking at how do we feed fungi and how do we encourage more fungi back into the system, because with a high disturbance like a fire, we've pushed it right back to the left-hand side and we're in there in this very bacterial dominated environment. So that fungi to bacteria balance, or bacteria to fungi, happens underneath grasslands. So we're wanting to push more fungal dominated, I imagine, for a lot of you, unless there are some of you here that are ranchers. So how do we push to get that balance so that we get good, nutritious, nutrient-dense grass growing that's palatable instead of some of those low successional weedy grasses. So things that bacteria like to feed on, they typically require more simple food sources. These are the green materials. So some of you who are proficient at making compost, you understand this. So the green materials are what make your compost hot. So green grass, green leaves, manure, and then I put some other things that we can use to just stimulate bacteria and get bacteria humming. So sugar, molasses, seaweed, fish emulsions. So more simple carbon foods are what the bacteria will really boom on. So there's things that we can do to kind of push for more life in the soil, but they're not necessarily going to feed your fungi. So I'd like you to consider that some of these early pioneer species, particularly in some of these grasses, will actually slow the biological succession process down. So it's not in their interest to have this process speed up and head into forest. So some of the invasive weeds that we have in the US, like knapweed, certainly cheatgrass, have different things going on in their root zones. So cheatgrass, for instance, has a very close relationship with archaea, so very primitive bacteria. It feeds those types of organisms specifically, and in doing that, it actually changes the soil environment around it to outcompete other grasses or other species. And as we know with cheatgrass, you then end up in a fire cycle. So it dries up, becomes great tinder, and on and on and on that cycle goes. So we're seeing that all across the Midwest now. They love these very bacterial soils. They're slowing the advancement of succession. if you want to ask questions as we go, that's fine. And if some of you will be highly trained in some of these environments, so you're welcome to contribute. No problems with that. So fungi, on the other hand, are also involved in disease suppression. They're also retaining nutrients. You will know many of them as decomposers, so they're breaking down. In that picture, you can see that's woody material. In this image, it's actually compost, that a cold composting process that we were doing. We're basically just using white wood chips, so they are poplar or cottonwood or birch or beech or elm, you know, like your English, or deciduous white fast-growing trees. These guys just, they feed fungi like nothing else. So this pile was actually made with just 200 yards of a white wood chip. We put in a little bit of liquid manure and added worms, and that's basically all that we did. And those worms got into those wood chips and turned it basically into to vermicast or worm castings. But you see the fungal hyphal strands in there. So there's your mycelial networks, right? So there's so many now that we can visibly see your fungi, because fungi are typically invisible. Right, so you have different types of fungi, some that are breaking down materials like that, and then your mycorrhizal fungi, which are the ones that are living in or on tree roots. After the fires, you may see this proliferation of the fungal fruiting bodies, so like morels or other morels. Did I say that right? Anyway, you'll see fungi that come up after fires, and you'll find the fungi foragers going out to get them right there. They're fruiting because they've just lost their host, and many of them can't survive without a tree, or if they can survive without a tree, they can't reproduce. So after that tree's gone, they're just panicking, basically. So you're seeing that last gasp, and they're setting the scene, hopefully, for trees to come through in future. Right, hopefully the internet's all right. It just told me that it was unstable. What fungi do is they form the macro aggregates. So the larger crumb structures is what fungi are responsible for. All right, so think of those strands as binding together and holding together these different aggregates. So the crumb structure is then bound by long strands of fungi. So imagine when you're walking through a forest, every footstep underneath you, every foot that you tread has about 300, so 200 miles of fungal network underneath, just what's underneath your foot, in that square foot of fungal networks, right? So huge amounts of knitting together. So if you think of that movie Avatar, if some of you have seen it, that idea that these forest networks are connected is very well established now. Those fungi are communicating from tree to tree and binding on and holding on to the soil environment. So really vital in our forest landscapes that we get these fungi up and running as fast as possible. They are also very important for yield and yield responses. They're very vulnerable to fire. Some of the fire measurements have shown that there can be up to 50% drop in the biomass. So we're not talking about individual species, but the actual mass of fungi can drop. As a consequence, we get an increase in erosion, we get a decrease in the mineral uptake. Remember I was saying about how they are releasing acids. These guys are the original mining companies, right? They're getting in there and they're taking off bound nutrients, if that's calcium or phosphorus, or your trace elements even. What we find is if the fungi are impacted like this, then there's a dramatic decrease in your water holding capacity, right? So they're holding on to water while they're also holding on to soil, right? It's also an effect of the loss in soil structure. So fungi are integral in building that soil crumb structure. If we impact on that, and we don't have those pore spaces, then we have a decrease in that soil's ability to hold on to water. Through fire, we also lose a lot of carbon. Carbon's role in the soil is to create a sponge. So we've got a couple of different factors going on with water, which I'll get into soon. So erosion like this can be common after hot fires. What we're losing here is that biological component. We've got soils that begin to slake, which means that they have no biological integrity. They lose the glues and the we call glomalin, the sticky aspects that actually hold a soil together get lost. So what you're seeing here is actually a microbial death, right? And so how do we stop this kind of impact happening? Because what we're seeing is not, and we're seeing this all over the world as we're going from super dry to flash flood to super dry to flash floods. Some of these rainfall events, you know, are getting more intensive, and we haven't got biology in these systems after a fire to really help that system recover quickly. So what are fungi like? They require what we call the more complex carbons, or the brown materials. So for the composters, these are the things that we're putting into a compost to stop it getting too hot. Things that are composed of high cellulose or lignin or chitin. So chitin is what fungi are basically made of, what insects are made of, and shellfish. So some people are actually putting, like around Salt Lake City, we're using brine shrimp, ground up brine shrimp to actually put out onto land to feed beneficial fungi. They're after your straw. We're using what we call fish hydrolysate. So fish hydrolysate is a complex fish product that's full of oils, it's full of enzymes, it's a very complex type food source, so fungi absolutely go nuts when you put it out. They really like it. Same with the humate. So a humate is a soft brown coal. So think about it like it's it's prehistoric forests. So it could be, 65 million year old trees and vegetation that over time has been worked on by different types of bacteria and fungi, and then under pressure has turned into these soft brown coal seams. So we're not talking about charcoal, we're not talking about what you can see right now after the fire. These are full of the biological signals, which we'll get into soon. They stimulate fungi like no one's business. Biochar, some of you might be familiar with this already. So it's a human-made material using carbon-based products with a pyrolysis machine. So under high heat, well, no, not under high heat, but cooking these woody materials without the presence of oxygen, basically, and you form these charcoal-like products. But they are different to what you're seeing out there with the fire, in that they don't have any oxygen in them. They provide a very stable structure for microbes to live in and on. And then the different types of wood chips. So some wood chips might be resistant to breakdown. So thinking your spruce, your pines, those, if it's got a very strong tannin smell, or, just have those beautiful, strong, oily smells, those smells are actually antimicrobials. Those trees are designed to resist the breakdown in microbiology. So if we're looking at feeding fungi, they're typically not the types of wood chips that we want to be using. And often if you put them out as mulches or try and use that as woody material to help soils recover, they can leach out some of those aromatic type products and create these water repellent systems. So I prefer to avoid those if I can, or if I'm going to use them, I'm going to let them break down for like 18 months and probably spray some fish on it just to help that wood material start to break down, and then use it. But I wouldn't use it fresh. Whereas the whitewoods we can actually use fresh. So what we find is it's not as black and white as that. We don't like, oh, we're going to lose all our fungal diversity and all our bacterial diversity. Some species will actually increase following fire. So this research has just come out this month, last month, yeah, showing that some bacteria are very, they're finding niches. So they're like, well, we can actually break down some of these materials. So you might see a boom in some of these populations and some of these fungi. So some of these species are actually pathogens, but they're booming after the fire comes through. So we lose biomass and biodiversity, and you can you'll have less biodiversity in some of these more narrow genera that may in fact be pathogens. Right? So we're not saying everything's gone and the slate's been wiped clean. There's biology in the atmosphere, right? So there's always microbiology moving around. One of the conditions that we see when soils get really hot is that they can become water repellent or hydrophobic. I was on an operation in New South Wales where we were seeing this in some areas where there'd been tree tree material, that got really hot. So have a look at this for yourself. Just put some water out, sprinkle it like that, or use an infiltration ring and have a look and see how long does it take for water to enter these soils. So if you've never done an infiltration test, you're basically just banging in a cylinder, make it 6 inches, and you're going to put in 1 inch of water and time it. See how long it goes, and then to put in another inch of water and time it. And it's the second inch that you want to time. That second inch needs to go in faster than 12 minutes, right? And 12 minutes might seem like a long time, and it is. But if it's gone slower than 12 minutes, your soil is totally broken down. Like your water system is really busted. It should go in quickly. Like in 30 seconds, that water should move through that soil. But you'll find if they've become hydrophobic, they're just going to sit there. So those hydrophobic conditions can be a byproduct of organic materials. So different types of plant species will have different amounts of these hydrocarbons or materials that turn into waxes. And so during the fire, they concentrate, they can move down through that soil profile, they wrap themselves around soil particles, and they can create pans. So we can see hard pans or impervious layers because of these hydrophobic, waxy materials that are coming from your organic matter. They can also be created by microbes. So when the microbes get super dry and super hot, they create waxy coatings as well. So we've got to look at, if you have hydrophobic conditions on your soil, how do we address that? Because if your soil is water repellent, it becomes your very first place to start. So I really encourage all of you to get an infiltration ring. And when we talk about infiltration rings, you can just make it out of a piece of down pipe or a steel ring, anything you can find, and just measure 1 inch of water and have a look at how well is it moving into the soil, because it's critical. If you don't have water, it's probably telling you that air isn't moving either. If we don't have air and water, nothing's going to go very well or move very quickly. So some of the things that we're using to manage hydrophobic soils is we're using low rates of alkaline type products. So they might be what we call the liquid limes or fine limes. So you think of the old whitewash recipes, which was what we call slaked lime or burnt lime. It's lime that is used in concrete manufacturing, or it's a byproduct from concrete manufacturing. It's a very soluble material. And you can also buy commercial fine limes. They can be a little bit expensive. So if you want to make a whitewash recipe, just Google whitewash recipes and make your own. They can fall out of suspension if you wanted to spray them. So yeah, you need an agitator typically if you're making your own, whereas the commercial fine limes should stay in suspension. And we're also using milk. So probably going on, you're crazy lady, but milk or milk powder, cow's milk, goat's milk, doesn't matter. What you find, what we have in milk is, yes, it's an alkaline product, it's high in calcium, but it's also high in sugars that feed beneficial microbiology. So in the atmosphere right now, all around you, you are now breathing in lactobacillus. So lactobacillus actually is in the atmosphere. You put milk out within minutes, lactobacillus will start reproducing in it. So it's a way of inoculating soils. We get that lactobacillus going just like in your gut system. It's a very beneficial probiotic, and it can be quite cheap. So what those alkaline products do is they disrupt that waxy coating. So yeah, you can either apply solid calcium, which for many of you in California, it's actually critical that you get some calcium on. So that could just be agricultural lime. That agricultural lime will help to chew up those hydrophobic layers, that waxy coating. I'm going to mention vermicast quite a few times because I'm a massive fan of it, but worm castings, if you think who's going to build soil the best, well, who's going to do it better than worms? So worms have been unchanged for hundreds of millions of years because they do the best job you can imagine, right? They produce the elixir of life out their back end, basically. So what comes out their back end, they have calciferous glands. The worm castings that they excrete have this bioavailable calcium in it, and it's also full of beneficial microbiology. The types of microbiology that are going to help, one, eat up these waxy coatings, and 2, stimulate beneficial microbiology to fast track the recovery of these soils. We can do seed dressings with those products above, so you can make up a seed dressing that might have a fine lime or vermicast. I've got some recipes at the end of this, and we will make this slideshow publicly available so you can share it and use some of these recipes, right? But we've got to address those hydrophobic conditions first if you have them. So vermicast for me is a vital fire recovery tool, right? So it's full of biology, and it's got these signals that are going to kickstart soils, right? And I know there are some good worm operations in Northern California. The marijuana guys know all about vermicast. They know that it holds onto water, that it helps grow very healthy crops. Apparently that's important. And yeah, holds onto nutrients, right? But it's full of this beneficial microbiology that's actually going to eat some of the pathogens that are now sitting in that soil. It's also going to help with the decomposition and detoxification of some of the heavy metals and materials that might be in the soil, right? We've got to look at how do we stimulate as much biology as possible to, oh, it's people asking questions. Oh, okay, I'm sorry. Now I see. Oh, questions are cool. Okay, so that's a good idea. So Henk is saying, what about gypsum? Sure. So gypsum, when we're talking about calcium, can also be helpful. So what gypsum does is it's a calcium sulfate. It's going to provide that calcium material. It's not necessarily going to buffer any pH, but what it does is it flocculates or opens up that soil. So flocculation means that if you've got these tight plates, then it actually opens it up. So gypsum is a good tool to open up soils. For some of you, what it can do is it can also flush nutrients out. And after a fire, I'm a little bit reluctant to be removing any nutrients because we really just want to get that system humming. But sulfur reacts with things like magnesium or sodium, and we'll take magnesium and sodium out of the system and replace it with calcium. So it can be a good tool. So yes, by all means, use gypsum. So vermicast contains a lot of the microorganisms which eat these hydrophobic layers. So Pseudomonas fluorescens, for instance, the Serratia and Bacillus species eat the waxy coatings. And I've noticed that actually there's a research project going on right now around using Pseudomonas to control cheatgrass. So cheatgrass does not like Pseudomonas. There's a lot of primitive grass species that don't like this specific Pseudomonas species. Pseudomonas fluorescens also eats the organisms that make frost damage. So these specific types of bacteria, it's actually another Pseudomonas syringae that makes ice crystals and frost damage on leaf surfaces. This Pseudomonas will actually eat those organisms up. So in that recovery process, it can be really helpful just to get a crop ahead. Right. So there are thousands of these signaling molecules in soil. So this is what is in your vermicast, is these signaling molecules. So healthy soil is full of your proteins, hormones, enzymes, and elicitors. So things to turn actions on in the plant or in the soil. In that soil and in the vermicast and in good compost, you'll have plant defense molecules. So salicylic acids, jasmonic acids, chitinase, and proteinase. So chitinase is the enzymes that plants use to defend themselves against fungal attack, and proteinase is what they use to prevent insect attack. Right? These defense molecules are stimulated by the different types of molecules produced by biology in the soil. So there's a symbiotic relationship between that plant root and different types of microbes so that that plant is able to get up above the ground and be healthy. So we call this process quorum sensing, right? So it's how plants and biology interact to either slow up or speed up turn biology on or turn biology off, which is called quenching. They discovered this originally in the 1960s, looking at how it was that deep sea, you know, like squid and different types of fish could infloresce. So they would suddenly have lights go on and lights go off, right? It's bacteria that are doing that signaling on and off. So they've discovered this process is happening in your body, for instance. So at any one time you could have strep throat and you don't have an infection, you're not sick. And then suddenly you've got a sore throat and now everything's gone virulent. What they found was the strep organisms would reach a certain plateau and send out a chemical signal to turn everybody on. So they've discovered that they could find the same signals to turn, let's say, cholera off. So they figured out if you can find what the chemical is that these biology are using to signal to each other, you can turn things on and you can turn things off. So it has the potential to replace antibiotics in the future. So instead of this broad spectrum, let's kill everything, it looks specifically at what is the chemical signal for different types of biology. So for us, we're interested more in how do we switch on as diverse amount of signals as possible to turn on that system again. So this is where a little goes a long way. So typically on rangeland, for instance, we're using like half a gallon of a worm extract to turn biology on. We can, so there are other products or things that you can make for yourself. So if you look at some of the Korean natural farming resources, one thing that you can do is actually go to remnant forests and collect some of that duff and material. So there's a process involving, cook rice basically, put it into the ground and leave it there for all those microbiology to get in there and start inoculating the rice, and then using that rice as your initial starter or your serum to then put back onto land that's been burnt. So I would encourage you to have a look at Korean natural farming as another tool for soil recovery. So our goal is to optimize this biological diversity, and getting that biomass optimized is absolutely critical, because plant health and nutrition is driven by all these different biological functions. And as we have more communities, there's more of these signals, then we have full gene expression. So what you find is if you only have a couple of different species, they can't switch on the genes for, let's say, temperature resistance. They can't switch on the genes for phosphate solubilization, right? They are very limited until you have an entire community. So we have all of that structure. You have the schools and the pubs and the hospitals, and families living together can now have a community that works. If you just have a few different species, that system's now in decline. We now don't have resilience. We don't have plant health. So we need that full community for full gene expression. And that's why we can look at just using foods to try and feed what biology's left in there, or using some of these inoculants. So I just want to show some examples of some of the places I've been involved in. So this operation here had a fire come through 18 months ago. This is tussock grassland. There had been some woody weed species, of which you won't be familiar with, so I won't mention them, that were burnt out in this fire. Not an incredibly hot fire, not hot like what you guys have experienced, but what they did host the fire program, and I haven't got the rates there because I haven't got clarity from a couple of people, but they put on the fish hydrolysate we were talking about, and they applied molasses and liquid lime. So this is looking at it's from a different angle, but looking at the recovery of that grassland, and you see the burnt shrubs there in the background, right? And so this is what it looked like 18 months later. So they had a researcher looking at doing treatments. So using that treatment compared to the controls. So the controls on the right that had no biostimulant. And then on the left, it's had the fish and molasses and the lime. What they were measuring in this case was just biomass, so dry matter. And so these areas that had had the treatment so there was a 5% plant yield recovery over time and a 74% increase in the treatment, right? So we're looking at yield responses specifically just from putting in these treatments. They are doing a DNA analysis now. So Dr. Peter Espy is doing this work in New Zealand. He's been doing a lot of work into how these different types of biostimulants are working. This work has not been published yet. So actions for remediation. We've really got to look at how can we kickstart these biological processes as soon as possible before a lot of these hydrophobic conditions kind of set in, or you end up with a lot of perennial weed species. So some of these, what we call perennial weeds, so long-living weedy species that may be seen as invasive, can be very difficult to move without, say, chemical applications, which we'd like to avoid, and can actually slow that succession back to trees. So it depends what the species are that are coming in. But yeah, what story we want to set for this soil? How do we want to set this environment up to recover faster? So the reason we use fish hydrolysate is it also provides bioavailable forms of phosphorus, nitrogen, sulfur, and these fishy oils which help to stimulate the fungi. So it's a really good fungal feeder. Just want to say I'm an independent. We don't sell any product. I'm not associated with any products. And the fish hydrolysate itself is a waste product, so they're not fishing to make fertilizer as such. But yeah, here's some of the products that they are producing. So we'll use liquid lime. So the reason for that liquid lime is just saying my internet connection is unstable. We were good. Right, these liquid limes, we're using them to feed a wide range of microbiology. Love those limes. From soil testing I've done sort of north of San Francisco up the Lost Coast, you guys have some real issues with calcium in a lot of places. Calcium is the foundation for health in any ecosystem. And it provides, if you think about calcium, teeth and bones, it's in every single cell wall. It is part of the important enzymatic reactions in plants and certainly in microbiology, and every single living cell, all right, needs calcium. So calcium, 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 in case you didn't catch that, all right? So it's there to feed microbiology, and it's going to break down those hydrophobic layers and just really help to kickstart the recovery of these systems. Anytime you are using any of these products, they need to go on with a carbon food. So if you're using lime, it's got to go on with some kind of carbon food material. So that might be the humic acids I was talking about. It might be seaweed. It might be the fish, for instance. But don't put lime on out by itself, right? We need to be feeding the fungi at the same time. And then molasses. I would be using a little bit of molasses just to get this system kick-started, right? Just to kind of wake it up, because it's been a real shock, obviously, for everybody and to everything. But your system basically just stalls. It goes into shock and shuts down. That molasses can just help to feed a wide range of biology. So the thing with fish and molasses is that it's very broad spectrum. We're going to feed a lot of different types of microbiology and get them going. The question is there an ideal order in which to apply these biostimulants? I would put them all together. These three things together would be fairly economic. You could put worm castings or compost extracts down by themselves. I think I'm going to mention them in a minute anyway. Yeah, so you want to have a fungal diverse compost or extract, so that's where we're taking a liquid off these types of products. Same with the vermicast, right? They need to be microbial balanced. There are a lot of commercially available composts. Do not use a lot of these. A lot of it's municipal waste type composting facilities. They're very large. They get really hot. They produce a lot of bacteria. But not necessarily, probably, if you test it, you'll find very few fungi in them whatsoever. So they're not fit for what you're trying to do. ask them for a biological test, you want to see that they're fungal dominated. Like this compost here actually has more fungi in it than bacteria. These are the types of compost that you want to be using. With worm castings, it's often challenging for them to get their fungal numbers up. We still want to see a microbial balanced vermicast. So it should actually almost look like this, like a lot of wood chip in it for the fungi to be growing on. If you're just applying compost for compost's sake, yeah, you could actually be setting your system in the wrong direction of what you're trying to achieve. One way to tell actually is to do a germination test. So get yourself some of your compost, put radish seedlings in it, have a look. Does it produce 100% germination of those radish seeds compared to, say, potting mix? If it does, that's one step. Then leave it out, just the compost, leave it out in the environment and look at what grows on it. Does it grow a whole lot of kind of gnarly broadleaf weeds and primitive grasses, or does it grow nice soft grasses and nice soft clovers? If it's growing clovers and soft grasses, then that's a good compost to use. If it's growing those other woody, those broadleaf weeds, don't use it, right? Because that's what it's going to grow when it's in your when it's on your land. So this is just a generic recipe that that I gave out to the group. It would be really great if we could get some people using this before I come. So I'm doing some workshops with Point Blue in May. I'm trying to think, in May. And so if some of these recipes have gone down, we can have a look at how has that actually changed some of these environments, and you know, how often are we going to be putting things on like this? You know, if you want to come in and get a recipe, it doesn't have to cost a lot, but just to get something starting to work. If you had the budget, you could double those ingredients, or you could do split applications. So do something now, yeah, and do something in fall, and also looking at maybe putting on solid lime applications, perhaps. I'm not going to say that without seeing your specific soil test, but I can imagine some of you do need solid lime. So yeah, that's just a generic recipe, but it's just designed to, let's wake the system up. You're going to need to make sure things are filtered if you're putting these through most conventional sprayers. So we use boomless nozzles so that we can spray things like worm liquids, but you can buy worm liquids already made up, so they're already filtered. So all of these products, if you buy commercial liquid lime, are triple filtered, so not triple filtered, they'll go through very fine filters. So this recipe should go through a conventional sprayer. There's someone on our list here today who's on the ranch that has this sprayer. I'm just showing it as an example of, there's different units you could hook up to try and get materials out. I imagine in places people are going to be pulling out logs and doing a lot of damage to soil with logging trucks and all the rest of it. So maybe you would, once they've cleared these areas, you could come in. And then these are some of the extractors. So people are actually putting compost and vermicast in this type of equipment. This is a company called Midwest Biosystems. And so these extractors mean that you don't have a lot of material that might block spray units. Yeah, and then this is a generic seed treatment idea. Well, actually, it's not a generic because I haven't given you rates, but it depends on the quality of some of these different products as to how much you might be using. About half a gallon of worm liquid to a ton of seed, so mixing that maybe in a concrete mixer, putting some fine lime in with that concrete mixer while you're doing it, some of the liquid humic acids, seaweeds, and again looking at that Korean natural farming, but getting something onto those seeds so that we can change the signal and actually set that plant up so that when it starts to grow, particularly if you've got the worm extracts, that plant grows, it's got the microbiology on it that's reproducing and in its root system, and we can start to kickstart that recovery. A lot of the treatments have been, putting seed out or putting just mulch out. They're not necessarily getting the response that they need to get. You need to set that seed up for success, because right now it doesn't have its microbial partners out there. And if it does, they might be like disease organisms. So by putting a bit of worm liquid, it stimulates germination for one. So the seed actually is communicating with the biology outside of the seed that's saying, hey, this is a really good environment, you should germinate here, and off it goes, and we set that plant up for health. So you will get better success rate with putting seeds out. Yeah. So really start thinking about it as more of that whole ecological, whole systems approach. We need to look at how do we create that environment for plant recovery. If you have water issues, how do we address water infiltration? If you're using mechanical intervention, so some people are using harrows or ripping materials to open some of these layers up, you must feed the microbes at the same time. So setting systems up so you can be dripping a little bit of fish or humic acid or whatever into those soils to help stimulate beneficial microbes, and particularly after heavy equipment comes out. Right, so it's those plants that are building soil. So if we can support optimal plant health, we do that with our seed dressings, and we do that with the bio sprays after maybe grasses and different plants are germinating. And I think some of you have had some moisture. Setting it up so the species that you're wanting to kind of transition to are the ones coming through instead of those woodier or weedy plant species. I just wanted to touch on, if I can leave it on that picture, I wanted to touch on some of the contamination issues. I guess it depends what those contaminants are, and if they're looking at actually coming in with machinery and just taking all your topsoil off. How do we then remediate? So fungi absolutely are key in remediation. They will actually, they can bioaccumulate, or they can turn some of these heavy metals back into rocks, which is really interesting. So there's geomycology, which is that fungi can actually take raw ingredients and turn them into stable rock forms, which is pretty cool. But we want fungi are the ones that are going to be really the ones that you want to be bringing in. So all of these tools will work to help stimulate beneficial fungi in the system. White wood chips, cardboard that's inoculated with things like your oyster mushrooms. There's a lot of research now into remediate or mycoremediation, so using fungi to help these systems get back into place. I'm with you, Terry, about not eating plants that are, vegetables and things that have been grown where there's a lot of ash and a lot of contamination, and it's something to be aware of. It's going to be hard to find material. Okay, so there's two questions. So when restoring hydrophobic soil, some research is suggesting not to mechanically break up soils to prevent erosion. In this treatment, do you recommend liquid treatments, a mix of solids and liquids, or what order of application if using liquids and solids? So, yes, let me go back. So, I personally, yes, I would not be suggesting mechanical breakup of soils because of this tendency to erode. This is where those liquid applications are very beneficial. They'll start working their way through that top surface. And again, if you can be doing seed treatments just to let that seed get down through, that will be the one that's going to help break through. If we're doing solids and liquids, then we want to do a solid mineral application first, and then spray your liquid on top of it just to help it move through. That's if you're doing those timings quite close together. So dioxins in particular, someone's asking, how might it accumulate in grass-fed pastured livestock and row crops? I actually don't know the answer to that, sorry. I have done a lot of work with dioxins. I don't know if there's someone in the group that can answer that. I'm going to guess that fungi are going to help with it, but I haven't looked into the research, sorry. So Bree's asking, if we already had germination, mainly those annual weedy species, would you recommend the same applications in seeding preferred species? Yes. So those annuals, you're going to get those quick clambering annual cover up really fast. That's great. They're trying to hold that soil together. Getting that recommendation out is just going to help push it past the soil condition that those species require. So you'll see specific species, like thinking of some of the thistles, for instance, they have anaerobic bacteria that live in and on their roots symbiotically with those thistles. So they require basically pathogens in order to survive. So by stimulating that soil environment and feeding higher fungal communities, then we make that environment less desirable for them and set it up for the species that you're wanting to put in. So yeah, I would be seeding preferred species. Again, it's going to, yeah, it can be costly putting seeds out, but if we're doing these seed treatments, you're going to increase their rate of germination. And there are some really good species that germinate without being drilled in. So I mean, if you're flying things on, but you guys need to talk to your local extension agents about native species that you might want to try, and I can't speak to that. So yeah, are there any questions? You can take yourself off mute if it's faster, if you don't want to type.
Hi, Nicole.
Hi, Sarah.
How are you?
Yeah, good, thank you.
You're doing a stellar job.
Oh, thanks.
I'm
It's always kind of weird.
Sorry, is it clear? Am I coming across clearly?
Yeah.
Okay. I just have I'm doing some research on Chilean needlegrass, and I've got a little conversation happening in a local. You may know Gyra, just north of Armidale. Anyway, big issues up there, and I've been doing a little bit of research and seeing how widespread it is in New Zealand, and I just wondered if this if you've tried this approach on it, you know, using the biostimulants and you know, exploring those possibilities, because it's a big disruptor.
Yeah, and I guess if we think about Chilean needlegrass, or which in some ways, well, it's not quite the same as cheatgrass, but it, you know, it has very shallow rooting systems. Look at what conditions does it favor and what does it not enjoy. And so if you look at some of the work around Chilean needlegrass, it doesn't like tall grass competition. So it's a management thing, as well as shifting it to that more fungal dominated. So it is a more of a bacterial dominated grass species. But yeah, I don't think these guys have Chilean needlegrass. So dig up roots of any of these plants and have a look at how do those root systems grow. Chilean needlegrass has roots that only go down about 2 inches. So the minute it's got good tall grass competition, it gets out competed. All right, I've got some written questions. Thanks, Sarah. Bree says, if we already had germination, would you recommend the same applications seeding preferred species between wood types where burned? So where there's pine, fir, and oak are being removed, what should be applied if these are chipped, mulched, and spread? So that recipe can go on top of that chip. So if you're chipping those pine and the fir and the oak, then using that same recipe is going to help break up some of those antimicrobials. You know, because there's still going to be some timber, obviously, that's not fully charred, and there's going to be the contribution. So I'd use that material as well as you could, and by putting on the fish and the lime, it's going to help prevent that hydrophobic condition that can form under some of those woody mulches, especially when it's dry. Max has asked, I'm wondering if hemp combined with native grasses could be an exemplar species to use with the biostimulants for remediation. Great pioneer to feed off that initial nitrogen post-fire. Absolutely, Max. Yeah, I think it would be a great use for hemp, particularly not it is a bioaccumulator. It will take up heavy metals. So as long as, and obviously it's not being used for food, but if it's being used for fiber, it can be cut and removed and taken off-site if there are contaminations. Yeah, and it will feed off that initial nitrogen that we have released post-fire. So you'll see there are specific plant species that will mop up the nitrates that we'll start to see formed in the soil. And this is another reason not to be eating food that's grown on these soils right now, is those nitrates are harmful for human health. They're carcinogenic. Microbes, and most plants don't like it, but there are some specific weeds that have the ability to mop up those nitrates. Some of your like your nettle family, your thistles, you think knapweeds, nightshade family, foxtail barley grass. So if you've got foxtail barley, it's often in there to help mop up a nitrate issue. So it's doing a job. We just want to look at how do we do it out of that job. So if we've got an excess of those nitrates in the environment, then those carbon-based products, so those humics and milk will do it as well, will actually help to mop up that excess nitrogen. But yeah, it would be interesting to see if people did want to grow hemp. Yeah, and hemp itself, yeah, it's quite, well, it's a weed. But yeah, once you've harvested that, starting to keep pushing those environments to more fungal if you're wanting to have trees as your ultimate climax. Okay, and any other questions? Oh, what would happen to mixed sawdust? What would happen if we mixed sawdust with newspapers, macerated it, and sprayed it on the ground to use the cellulose out of the paper? Yeah, that's a good idea. Again, I would still be putting something on that's really, it's going to provide some phosphorus. So your fish again. So you could macerate and mix it and put something like seaweed in it as well. So seaweed's got a lot of trace elements, feeds a lot of microbiology. But yeah, that could possibly be quite nice. Just got to watch, some of these, we are still quite dry in places, and, not having layers form between some of these mulches or paper pulp or anything that we're wanting to put out there. Hi, Jeff. Your question probably doesn't relate to this fire webinar, but you can ask me that at another time. There was a question here. After helping the soil, do you think we should help plants to grow with foliar feeding to help feed the microbes? Yeah. So, Tant, kind of what we've been talking about. So that recipe, just to get you started, there's lots of other things. So there's the effective microorganisms, some of the Korean natural farming, the lactobacillus serums. There are products that we can make using our natural inoculants. So instead of having to buy a lot of products, there's a lot of things that we can make and just spread out there to help stimulate microbiology and then inoculate it with native organisms. So Beth is asking, can you explain further what kind of contamination happens due to fires? There are a lot of heavy metals concentrated. There's been a lot of, obviously, houses go up, there's been vehicles go up, so we've got hydrocarbons, we've got dioxins, you've got plastics, you've got, you just think of the stuff that's in your house setting on fire. So a lot of materials that are carcinogenic, a lot of materials that are heavy metals, so they're going to cause problems for human health and animal health. And this is why I guess the regions are talking about scraping soil and removing it. We can remediate it. It's going to take time, and as it is with all of this, it's a time process. But there are things that we can do to speed it up. So Jeff Smith is saying, Henk, your mix will drag nitrogen from the soil to break down sawdust and paper, especially if you use it thick. Yeah, so that's why we're thinking about adding some other things. So some kind of nitrogen-based materials, the fish, the seaweeds, to help balance that carbon to nitrogen ratio so it doesn't pull nitrogen from the soil. That's why I like the whitewoods as well, because they have a good balanced carbon to nitrogen ratio already. So Megan's saying, how do you flocculate compacted soil without compromising fungal integrity? So that flocculation process, it's a chemical process, but it's feeding calcium and sulfur with the gypsum. So this process of just opening up will actually stimulate fungi, not suppress it. Don't go and, some people are putting like 5 tons of gypsum on. Don't do that. We have this tendency to want to hammer things and make things move faster, and actually it sends your system in the wrong direction. So if we're using gypsum, you know, maybe you're going to use, I'm not going to give you numbers because it's a soil by soil basis, but you're just putting enough on just to open up that top 1 or 2 centimeters, and it's actually going to help to stimulate the fungi in that process. So yeah, good questions. Does anyone want to ask one? Oh, so Emily says, do you need to do soil lab tests to check soil is okay after remediation, or can you tell by looking at plants and insects? Emily, I think with the types of contamination that you're looking at, you want to get your soil tested. You want to know, because your plants might not necessarily tell you that there's been some major heavy metal contamination. And I know of operations where maybe they haven't been supporting microbiology as well as they can, and having heavy metals coming through to the plants, and then having some major health issues. So I wouldn't rely just on the plants to tell you, or the insects. They'll tell you a lot, but they're not going to necessarily tell you that, say you've got a cadmium or some kind of heavy metal that's sitting in that system. So yeah, please get soil tests done, particularly if you're going to be growing food. Can you add stimulants like comfrey extract or yucca? Absolutely. They will be helpful. Those yuccas are good stickers. They also feed bacteria. I just find they're a bit expensive. So yeah, there's a multitude of different products we can use. The comfrey extract, if you're making comfrey extract in your own land, then absolutely put that out there. It's got a lot of silica in it. It's very beneficial for microbiology. Yeah, I'd add, if you've got those materials, you could add that to that general recipe. Yeah. Cool. Any other questions? You guys have been awesome. It feels a little bit like I'm sitting in outer space talking to myself. So your questions make me feel like, oh, there's humans out there. All right, Bree, is there anything you want to add? You can talk.
I just want to say thank you very much. Really appreciate you putting this together for us. And thanks to everybody for joining. We hopefully got it recorded, and so we'll have it available. And then I think, Nicole, you said that the slides would be available too.
Yeah.
And I know there were some people that weren't able to join because of conflicts with time. So hopefully we'll be able to get it out to people who weren't able to get on right now with us. So big thank you. For the California folks, we are, like Nicole said, she'll be out here in May for some workshops, one up in Redding area and then one in the Susanville area. So if you're interested in those, and spending a couple days with Nicole, more focused on production topics, but you can certainly email me to get more info on those.
You
Yeah.
were going to make some recipes? Some recipes?
Oh, yes. So I will, the fire recovery recipe was in the slideshow, so we'll send that out. And also, yeah, I just had the fire recipes in the slideshow, so we'll send that out. We'll make it available. Okay, and someone's asking, can we get a transcript? How do we do that, Bree? Can we do that? I don't know how we do that.
I'm not sure. I'll have to look into it.
Find a transcript that understands the New Zealand accent. All right.
So you were talking about a whitewash recipes and things like that you mentioned before.
Yeah, so the whitewash recipe, it's just slaked lime and water. So it's a slaked lime or burnt lime is what it's called. You get it from the concrete operations, concrete factories. They sell it. And not calcium chloride, right? It's a different type of calcium, burnt lime. And yeah, and so you can just find those recipes online. Just Google whitewash recipe. It's pretty simple. Yeah. Oh, hi, Robin. Didn't know you were on here. That's cool. There's some awesome people on this. I'm really, yeah, I'm delighted that you've managed to join us today. Yeah, the whitewash recipe. I'll give a couple of recipes to Bree. Where will you share this, Bree? Will you put it on your website, or shall I put it on mine?
I can, yeah, I'll probably put it up on like a Google Drive and then email it out to folks.
Okay, just thinking of some people that couldn't attend.
Yeah, if you have it on your website, I will certainly direct people that way.
Okay, all right. We'll make sure that everybody that's participating gets the slideshow and the recording, and yeah, we'll see what we can do to make it publicly available. I think more and more people are dealing with fire and catastrophic fire across the planet. Even New Zealand is having some catastrophic fires. So I think information like this needs to be publicly available. Yeah. Cool. All right. Well, thank you, everybody. Really interesting questions. Thanks for coming along. And thank you. See you later.
Bye.
Bye.